Hi, I'm John Gagliano, Senior Director of Biopharm Catalyst.
Hello, I'm Chef, known as Chef Station, a 20-year vet in trading biotech stocks.
And this is Biotech Bulls and Breakthroughs. Biopharm Catalyst is not a registered investment advisory firm, and the views on this podcast are not intended to be financial advice. Guests on this podcast may hold the stocks discussed in personal or other investment accounts. This content is for informational purposes only and should not be considered investment advice. Always do your own research before making any investment decisions. If you find the information on this podcast useful and would like more Catalyst information, Biopharm Catalyst is your premier resource for Catalyst data for over 1,000 publicly traded biotech companies. We track clinical trial-based catalysts, everything from IND filings to drug data releases for phase 1, 2, and 3 trials to regulatory decisions. Our Catalyst calendars are updated as soon as the press release comes out, so you can be sure you have the most timely and accurate information to inform your biotech trading. All of our plans come with a 7-day free trial, and we frequently run discounts on our platform. So check out www.biopharmcatalyst.com to get started today. Good morning, good afternoon, good evening, depending on where you're listening in from or viewing in from. Welcome back to another episode of Biotech Bulls and Breakthroughs. I'm John Gagliano, Senior Director of Biopharm Catalyst. And today we have the pleasure of being joined by Neil Lineberry, VP of Corporate Development for MoMA Therapeutics. So I know a lot of times on our podcast, we have, biotech investors, Chef, talk a lot about the markets, but I'm really excited to actually have Neil from, an actual biotech company to give a little bit of a different perspective on the market, on some of the innovative things that they're doing, just to give folks a little bit of a different viewpoint and kind of a lens more onto the science side of the equation. And also, to be able to identify some opportunities directly from the source of one of the biotech companies that we represent. So, Neil, thanks so much for being on and welcome.
Pleasure, John. Thanks for having us.
Absolutely. So I guess just to start for everyone on the call and everyone viewing in, could you just give us quick background on yourself, kind of how you got to be part of MoMA Therapeutics and VP of Corporate Development, and just a little bit of your background?
Happy to. It's a bit nonlinear, so hopefully I'll make it clear. But I was a trained immunologist in grad school, and towards the end of that, it became pretty clear that there were many ways that I could affect drug discovery and bringing new treatment to patients rather than just being at the bench. So I explored potentially founding a biotech, wrote a business plan with some friends and pitched to VC, but I didn't really feel like I had enough content or I had enough direction. So I really wanted to get some training. And that's where I decided to go into life science consulting here in Boston with Leerink Swann, which is a very unique area because they're one of the first life science only investment banks, but they had a consulting arm. And what they did is that allowed us to work with a wide diversity of clients, whether it was pharma or biotech, medtech, pharmaceuticals, et cetera. And so after I went through that and kind of did the standard consulting growth thing, I wanted to actually get a bit closer. And so that's why I joined Takeda. And I was the strategy lead for a new therapeutic area in GI that they were forming around Entyvio and some other assets as well. And then that went for a couple of years, and I said, I need to go even further kind of down this tunnel and get really hands-on. And so I became a program lead on the oncology division, which kind of leveraged the immunology background that was really coming with IO and next-generation IO with oncology treatments. And so I was able to get a drug approved in colorectal cancer, applicable INDs, and really kind of tunneled in. And then I made the decision in January this year to say, you know what, I actually want to join this biotech party and see what this is all about. And so I was able to come in at the corporate development level, do a little bit of strategy work, but also help the teams thinking through how they're doing development plans and whatnot. So I've been here for about 9 months since the start of January.
Awesome. And so far, so good, I imagine.
It's been an amazing journey so far, incredibly different than what I've done before. We'll talk a little about how the markets have influenced some of that, but the company here has been really amazing with welcoming me and also the progress we've made. And we're coming up that we'll talk about on some really exciting inflection points that we hope will be able to give some clarity about some of the molecules and classes we're working on.
Yeah, absolutely. Well, that segues nicely into the second question is just, just a little bit of background on the company, how it got started, what the general mission statement is, and the general approach to drug development.
Absolutely. So MoMA was founded a little over 5 years ago out of the labs of Dorothy Kern, Evan Nagalis, and Johannes Walter, along with co-founder Timur Yusufzai. And they were funded by Third Rock out of Fund 5, that also syndicated with Nextech and Cormorant in the Series A. And then Goldman Sachs Asset Management came on in Series B. And the whole purpose behind the company was to really crack the nut on difficult to drug targets, specifically proteins that are highly dynamic. And this hadn't really been done before. And so our whole goal behind this is to be able to create a whole suite of different avenues and approaches across not only oncology, where we're focused now, but also potentially in other therapeutic areas to really hone in on these areas that have been overlooked and underserved to date that we feel could be able to bring the next generation of therapies to patients and specifically be able to move the overall standard of care.
Very cool. And I know that you have a pretty large focus on precision medicine. For folks on the line who might not be as familiar, what exactly can you define? What exactly is precision medicine?
Yeah, a precision medicine. It's a big buzzword. It's kind of thrown around, but it really, when you look at it, is all about tailoring therapy to the underlying characteristics of the patient. In oncology, the one that's gotten the most attention, has had a lot of development because it's been fairly successful to date, has been this is focusing on driver mutations in tumors because these are the ones that ultimately are leading to the growth and the metastasizing of the tumors. And so when you really begin to tailor in on them using molecular approaches and other approaches that have been developed over the past 25 years or so, that you can really get an idea of the underlying pathways and drivers that are putting those tumors into place. And so the classic hallmark of this was Gleevec, which was approved in 2001, and it focused on a particular mutation called BCR-ABL in a heme tumor called CML. And that kind of started the stampede of saying, hey, look, this could actually work. And when you combine that with sequencing the genome, in addition to other technologies like next generation sequencing, et cetera, it's just had a massive explosion in our ability to really think about, identify these pathways, and then design therapies to shut them down in ways we hope that be able to prolong the patients on treatment and prevent resistance.
That sounds awesome. What in terms of from what what I know about MoMA, there also seems to be a pretty big focus on dynamic proteins. Can you tell us a little bit more about what that means and sort of how that weaves into what you just mentioned in terms of what you guys are working on?
Absolutely. So when we say dynamic proteins, we mean proteins that use energy to affect motion or work. And so some examples of this are transporters, vesicular traffickers, and things that are involved. A lot of it is in DNA replication. So how you actually read the DNA, how you orient it in a certain way, and actually replicate it and put it back into place, right? So this is a wide class of proteins, over 400 that we've identified so far and continuing to grow. And so the challenge with this is that these proteins move very, very fast. And so to be able to understand the actual conformation of the protein that it actually adopts can be
Yeah.
very challenging. The analogy I always say is that if you kind of went through physics and you went through a point where they say, hey, an electron is a cloud, but it's also a particle, and you can never really know where it is. And I'm sure for those who went that class were kind of like, what, huh? What does that mean? But in
Right.
reality, it's actually true with these proteins because they move so fast, you actually kind of don't know what conformation and using the current techniques that we have. So what we're able to do is we're actually able to lock the proteins in certain conformations and then actually do the cryo-EM and the structure-based work around that. And predicting that takes a lot of work. It's a lot of computation that we have, and it's kind of an iterative cycle. And it hadn't really been done before until the company was founded, and a lot of the founders had developed this technology outside this at the Howard Hughes Medical Institute. So that's what we mean about our overall platform, what we call Nomatic, and how we're able to target these proteins, which have been a bit frustrated researchers up into this date because they didn't have these techniques to really find the right conformations of proteins, predict them, and then find the druggable pockets in them.
That's super helpful, and it sounds like quite a bit of innovation going on, especially since, correct me if I'm wrong there, but it sounds like you guys are sort of pioneering this to some degree.
Yeah, I think it's the combination. So the individual units we have around identification, target ID, and the screening capabilities we built, in addition to the structure work that largely came out of the founders, in addition to the computation, putting that all together is really kind of the secret and special sauce to MoMA. Others may have bits and parts of it, but it's really how we integrate that together and then focus specifically on these highly dynamic proteins. And while we started with ATPases, we'll talk about the pipeline in a minute, we've
Yeah.
also been able to diversify outside that into other proteins. And then, ironically, there's also some proteins aren't dynamic per se, but they have elements within them that are dynamic. And because of that, we've been able to further kind of get into those classes as well, beyond just what you classically think with ATPases.
Yeah, absolutely. That makes a lot of sense. And so I guess that kind of brings us to, what you guys actually have in the pipeline. Can you tell me just a little bit about your current pipeline, what assets you're working on, what indications you're targeting? Just kind of give us a brief overview of that.
Happy to. So our lead program targets DNA polymerase theta. So this is part of the molecular gene family that is involved in DNA replication and repair. And so this is actually a backup pathway that's rarely used in cells unless you have deficiencies in homologous recombination, which is by far the biggest pathway that's used to repair DNA. So that can happen with specific mutations that make it deficient. So we call it homologous recombination deficient, or HRD. Now, there has been a class of proteins that have been approved for this since 2014 called PARP inhibitors. And what PARP inhibitors actually do is they take advantage of tumors that have deficiencies in the space and kind of make the DNA damage persist and get bigger such that it leads to tumor cell death. The challenge with PARP inhibitors is that they lead to very quick resistance in many tumor types that leads to very short duration of therapy. And so what happens, what they found out subsequently since then, is that one of the mechanisms of resistance involves an alternate or backup pathway DNA repair that's mediated by DNA polymerase theta. So our idea is to be able to have an inhibitor of DNA polymerase theta on top of PARP inhibitors in the context of this homologous recombination deficiency, and we're able to disrupt the market and bring much better therapies with less resistance, because we're targeting the primary form of that to patients who have these particular tumor types. The most common patients who have this are breast cancer, ovarian cancer, prostate cancer, and pancreatic cancer, but there are also some others as well that we potentially could expand into. So that's our lead program. Our second program targets a very different part of the overall DNA repair pathway, but it is still very profound in the tumor types it is in. So this is inhibiting what's called the Werner helicase. And again, this is a pathway that's not often used in cells that are normal. But in tumor cells, what happens is sometimes you get expansions of parts of DNA called TA repeats. So these are two nucleotides that are expanded because they're not able to replicate properly. And when that happens, it causes the DNAs to adopt really funky conformations that are difficult to replicate. So it's like you create a big knot in the strand, and so it's the polymerases and the other things aren't able to get through that. So Werner helicase comes in, it specifically unknots things, which I really wish I had when I was in Boy Scouts back in the day, to tie the knots and unknot everything. That would have been really helpful. And so this is really used in these tumor types that have these mutations that cause these knots and secondary structures. So our thought, again, is to target this. And this would be the first targeted precision oncology therapy for these mutations called DMMR or MSI high. The standard of care is actually using immunotherapy, but this is actually targeting the underlying tumor driver. And then fundamentally, we think we can actually combine them together and have a really profound effect on these patients. So those are our clinical programs. In discovery, we have a whole plethora of different things that are also focusing on novel targets or novel takes on targets that we've known so far. And they're already, they're from hit validation all the way through lead op and DC candidate stage and whatnot. So it's a bigger diversification, kind of, burrowing further into this hole, especially with novel targets that haven't really been seen yet. As I alluded to previously, some of them are actually, moving outside of ATPases into other areas, and also it's almost like little pockets within proteins that have highly dynamic areas. But the whole protein itself, you wouldn't think is dynamic. And a good example, I'll just make something up here, is like kinase, right? You think it's in signaling, it's phosphorylation, but there could be disordered regions that are highly dynamic within that. And so that's kind of the next frontier that we're focusing on as well. I would be remiss if I didn't finish talking about our two collaborations that we have, one with Roche, one with Bayer.
Yeah, I was going to ask about the Roche. Yeah.
Yeah, so these, again, Roche is a multi-target collaboration, almost 2 years in. Bayer is a very specific collaboration that we have. And so this is a great not only validation for our pipeline, but it being able to tap into their resources in terms of libraries they have to screen, in terms of some of their thoughts about how they do things, and just general overall, they have some big fancy machines and databases that we don't have. It's really helpful to kind of take our platform to the next stage.
Yeah, absolutely. No, those are significant collaborations. I was reading a bit about the Roche one for sure. And then you guys have something. It's called. Are you guys able to explain a little bit about your knowledge based platform?
You talking about Nomatic?
Yeah, I'm sorry. Yes, Nomatic. That's what I meant. Yeah.
Yeah, exactly. So Nomatic is kind of the core platform, discovery platform within MoMA, and it's really made up of 4 parts. The first part is target ID and how we do screens. We'd be able to do some fairly novel things with the screens, whether they're covalent screens, whether they're fragment screens, or what have you. The second piece is once we actually identify the proteins of interest and show they have strong genetic validation in tumor types or disease, then we move into the structure function area to be able to, as I mentioned before, find those confirmations that are realistic of what's happening in the cell, lock them, and then find the various ways through SAR and other techniques to say how are we going to find the pockets and actually drug them, and then to optimize the chemistry around that. And then we get into computation. This really works synergistically with the structure, is that you're able to iterate using strong approaches. We have a little bit of AI in here, not huge, but a little bit that's working there. And it's really kind of a back and forth between structure, function, and computation to be able to understand exactly how do we optimize against the binding pockets that we see. Sometimes the binding pockets are cryptic and no one's ever described them before. We have to figure that out. So it takes a pretty big computational approach. The final piece is once we actually have an idea of the inhibitor that we're going to use, then we have to understand what's the patient type we're going after, how are we going to accelerate and tailor development, getting back to the precision oncology piece here. And so we've been able in some of our clinical programs to put some novel kind of companion diagnostic-y kind of patient selection criteria in there to help enrich the patients we think are going to respond. And we've been able to tailor how we do our development approach, whether we're thinking about specific sites or regions or inclusion and exclusion criteria to bring into play. And so that's really going to help us narrow down the patients that are going to respond, run the trials very quickly, get the results, and then move on to later stage studies. That's really the essence of the Nomatic platform. And as I mentioned, we kind of putting that all together in one place with people who have been here, who are world class and have been here for 4 to 5 years in some cases, is really one of the differentiating factors for MoMA.
That's really interesting. Thank you for walking me through that. Just getting back real quick to the pipeline. Obviously, a lot of the folks that use our platform are interested in upcoming data readouts, regulatory decisions, et cetera. Do you have any expectation around some of the timing of important readouts for some of the assets you have? Some of the assets that you have, and where these programs will go next.
Yeah, this is a critical moment and upcoming time for MoMA, and it's really exciting because all the work that's been put into identify and crack these novel and very difficult to drug targets is coming to fruition. So our first program, DNA polymerase theta, which is also called MoMA 313, is gone through dose escalation for about 14 months now, and we expect that we'll have an early read by second quarter of next year. In addition, the Warner inhibitor, which just went into the clinic this summer, MoMA 341, we anticipate, again, we'll have an early read about the same time next year. And the reason why that the timing is a bit accelerated for that is that we're focusing on monotherapy activity there to understand what the molecule can do. Whereas MoMA 313, as I mentioned, kind of plays in the PARP inhibitor HRD space. And so that's more of a combination play. And so that takes a bit more time and capital to be able to get there. And that will be an important point we'll talk about later when we think about the overall biotech market and kind of some of the trends that are happening there. On our discovery pipeline, our collaborations, we've been able to get milestones on them to show progress and productivity, and we're moving those towards stages where we potentially could be, making some significant progress on candidate, moving IND enabling studies and whatnot. And so these are all things that are coming together for us where we can hopefully play with an improving biotech market that will be able to have these inflection points and continue to inflect the company's growth and move even further into development, bringing these therapies to patients.
That's great. Sounds like a lot of exciting things happening in the near future here. I guess in terms of your competitors in the space, like who do you view as some of the main competitors, and what would you say that MoMA's competitive advantage is?
Yeah. So first to say, there are many people in the space. This idea of kind of synthetic lethality we're playing on, in addition to difficult to drug targets and dynamic targets, has attracted a lot of interest, which is great because to me, it's all about having to bring finding this innovation and bringing it to patients. So to me, I congratulate all of our competitors on this, and I wish everyone the best of luck to be able to do that, and hope we're all successful because that's the ultimate outcome that really
Yep.
makes a difference, right? If you had to target specific companies, IDEA has had some play in this space as well. In addition to Repair has been in the DNA polymerase space. There's a number of others there as well, and that's kind of a big spread in terms of the companies and where they are and what they're focusing on. But I think we all view this as like it's rising tides lifts all boats here. In terms of our differentiation, kind of referring back to our platform, what we built, it's really around those four elements of the ID, structure, computation, and the biomarker and the translation, bringing that to the clinic. But I'd be remiss if I didn't highlight the people that we have here that have some, as I mentioned, have been here for many, many years and kind of really, as you talk about the knowledge base, it really exists not only in the computers and on the cloud, but also in the
Yeah.
people that are here as well. And then I would also want to make sure to give a shout out to our founders and to also our investors. There's many more investors in the city beyond the ones that I mentioned, and they have been universally supportive and constructive in terms of the conversations we have. And that is often not talked about, but when you have a healthy relationship with the ecosystem and the board and the investors, it makes things easier when you're trying to do really challenging things like crack novel dynamic protein targets.
Absolutely, without a doubt. No, that's great. I and I had a question just related to opportunities from the innovations from an investment standpoint, but I feel like that was somewhat covered by what you guys have going on, and sort of the breadth that you're working on and the various assets. So I think for any listeners who are on the call, I mean, there definitely is opportunity in this space. It sounds like. I guess just kind of one final question. With where we're at right now, in the world and in politics and healthcare and innovation, how are you feeling currently about the biotech market as a whole, about your space and the ability to innovate moving forward? Are you optimistic, pessimistic, neutral? You think we're heading in the right direction? Anything, any things that you would like to see happen in the future to sort of foster that growth?
Yeah. It's a good question, John. I've only been for about 9 months now, so it's not has a long history like others like yourself. And speaking of dynamic, hopefully by the time this comes out, that things haven't radically changed because it seems
Yeah.
like. But I would say that fundamentally, I'm in a space where I'm moving from neutral to cautiously optimistic, and I'll give some reasons why. Obviously, with the caveat that no one can predict the future, so this is just little old me kind of putting my finger in the air and trying to integrate some things together. But fundamentally, what I've seen is that early stage investment hasn't really changed that much. Some of the rounds have gotten a little bit smaller, but the idea of like seed rounds and A's, and importantly, the venture capital funds are actually raising these rounds that focus on this, like Sanofi Ventures, Vivo, et cetera, have been kind of chugging along. There hasn't been too much of a difference there. You see a lot of this happening because while it's a high risk profile, the capital burn that they take to do this is generally much lower. And there's a lot of excitement. You always want to have novel innovation. It's people are generally want to see this happen and say, "Oh, that looks really cool. I want to be able to do that." Right.
Yeah.
What really has been the issue is on the other two levers, which is around later stage private biotech and then the public biotechs. And fundamentally, this is not new. The post-COVID kind of boom in biotech led to what many have characterized as too many public biotechs out there that aren't truly going to be able to bring the innovation that you need, right? And so
Hmm.
the fields had to recognize that and work through that. And there's some mechanisms for that, but similar to kind of like starting a trial and ending a trial, there's a lot of celebration about starting. There's a lot of mechanisms for that, but ending a trial or realizing it's futile is often not as well explored, and it can be very difficult. Same thing in the life of the biotech, being able like, what do we do with this? Should we keep going? Should we not? And what form should we do? And so that's why you kind of see that kind of have to flush the system. And so you've
Yes.
seen some examples of that, but it hasn't kind of totally gotten there. But there have been some progress, right? The other thing on the public side that's been very helpful is there have been some takeouts. Whether you're talking about Maris or Metsera, Edinon Bio, Verona, Blueprint over the past couple months, these have been pretty substantial. They've been pretty high numbers on them, so it'd be nice if there were some little tuck-ins, some singles and doubles in addition to the big homers and grand slams that these represent. But that represents finally capital being able to be recycled back a little bit from pharmaceutical strategics. We also had an IPO a couple weeks ago, so that was good to see. So it seems like that the public biotechs are starting to move a little bit. The reason why I bring it up is because the late stage, kind of clinical stage private biotechs, we're kind of at the back of the queue when you think
Yeah.
about a capital investment, right? Because we have the highest risk profile. Because even though we love DNA polymerase theta and we're in helicase, we think it's going to be amazing for patients. At the same time, we don't have the clinical data to back that up. So people are like, "Hey, I don't really see validation quite yet, but I like what you guys are doing." And then we spend a lot of money to be able to make clinical trials go. So we're kind of in that 2 by 2 in kind of the worst area with the least validation with the highest cash burn. And so that means that unless there's really confidence in the investing sector and these other areas are working with early stage investment, IPO takeouts, that it's just not an area where people are going to go to yet. However, there was a private takeout with Capstan by AbbVie, and one of the reasons why I think that that's very interesting is that AbbVie said we're going to take the risk on a relatively unvalidated target because the innovation is so amazing it could really change the standard of care for how you do CAR T therapies for oncology or autoimmune conditions, right? So they said we're gonna get over the kind of inherent risk profile. We're going to believe in this and take it out very early. So that says to me that it's people are kind of coming around. That the engine is starting to work and be lubricated a little bit, but it's going to take a bit of time. I don't really see this as being like a lightning in a bottle and things are going to just like jump off the page. And so that's where I'm kind of neutral to cautiously optimistic is where I'm landing.
I think that makes a lot of sense, and I would probably agree with that sentiment on our end and everything that we're hearing from investors. I mean, I think that to your point, it's a very dynamic world we live in, and anything can change at any moment. But I do think that there is a lot more optimism moving forward for some of the innovation, and so far, at least from our perspective, there's been a lot of opportunity in the biotech markets year to date, and we think that that's going to continue and even be better in 2026. So we would definitely agree with that sentiment. But Neil, I think that's all we had time for today. But again, I really appreciate you coming on and having this discussion with us. I think it's extremely helpful for our audience to just hear from kind of the horse's mouth as it comes to drug innovation and drug manufacturing. And like I said, a lot of times we have more of the investment based folks on, but I think hearing about hearing from the actual companies that do this is extremely important and provides a lot of value. So I really appreciate it.
Happy to, John. Thanks for bringing us on and seeing from the biotech perspective. And if people are really interested in the markets, I really recommend people check out the ESMO conference coming up in a couple weeks because there's going to be some data disclosures about Paul Theta and Werner in addition to some others. So that's going to be a really interesting time for us. And hopefully it's giving us a tailwind and we can continue to push forward in the clinic for these readouts next year.
Yeah, absolutely. I mean, certainly a lot of our audience is hyper focused on the medical conferences, ESMO being one of the big ones. So we'll actually be having a dashboard. I should say we actually have a dashboard currently out for some of the readouts that we're going to continue to update. So for folks on the line, definitely check that out on our website and stay tuned for more on ESMO. But again, Neil, thank you so much, and I really appreciate the time. And for those folks listening in, If you like this kind of content, please let us know. Please hit the like button, subscribe, leave a comment, let us know on our contact form, and we can do more sessions like this. But thank you so much, guys. I appreciate it.
Thanks, Sean.